This morning we have 5 short presentations covering a range of subjects. The first 3 are agronomy related, after which we'll pause for approximately 15 minutes for the interactive Q&A session. After that, we have 2 presentations which are related to our research strategy to build IPM-based approaches to support the sector in advance of future farm to fork goals. Again, you'll have an opportunity after these 2 presentations to interact with the speakers through the Q&A session. So without any further delay, let's get started. And I would like to welcome Sara Tudor, who is a Walsh scholar based in Aberystwyth University in Wales. The title of Sara's talk today relates to designing management practices to enhance both yield and grain milling quality of winter and spring oats. So over to you, Sara.
Okay, thank you. Oats are the 3rd most popular crop, cereal crop, grown in the UK and Ireland, with planted area, production and consumption increasing yearly. Due to this, there is also a greater demand for milling processing. Therefore, producing grain to meet milling specification is key. So in addition to yield, it is also important that growers consider the quality of the grain too. But there are many factors that can affect yield and quality, such as environmental variation, variety, and nutrient inputs. We all know the importance of nitrogen for crops. However, current nutrient guidelines focus on achieving optimum yield and do not consider the effect of N on grain quality. So the challenge, and there is also increased emphasis on reducing N inputs. So the challenge is to increase the competitiveness of oats, minimizing environmental impact, and maximizing the benefits for growers and milling industry by achieving optimum grain yield and milling quality. My aim is to deliver a guide for best practice in agronomy, pre-harvest and post-harvest crop and grain management to improve both yield and milling quality. To do this, I'll be looking at the effect of different levels of N on crop growth and development and on yield components and quality of winter and spring varieties. I'll also be analysing the relationship between these characteristics. Other factors include looking at how maturity, time of harvest, drying and storage regimes affect milling quality. And I'll also be using image analysis to gain an understanding why some oats are more resistant to dehulling, which reduces milling efficiency. I've been collecting data and examining over 20 traits. These include measurements and assessments for crop growth and development, which were taken at specific growth stages throughout the season. Different yield components were determined from pre-harvest tiller samples. Here are the grain quality analyses carried out to assess the physical and chemical composition of the grain. The top right image is a snippet of a grain image analysis scan. Advanced improvements in grain image technology enables me to analyze the morphological features of around 15,000 grains a day. The bottom right are electron microscopy images of whole oats, which is used to examine the attachment of the husk to the groat. Initial images showed cracks at the base of the husk of a variety we know dehulls well, but these cracks weren't present for a variety we know is difficult to dehull. Developing a better understanding of the relationship between all these traits, as well as how they respond to N, is vital in developing the management practices. So far, I have yield component and grain quality data for winter and spring oat trials, shown in these tables, which were grown in different countries of the UK and Ireland. All varieties involved are from the UK recommended lists, as most of these trials are part of a greater UK oat initiative. But Husky has been added to this season's trial at Carlow. All trials include 6 nitrogen treatments ranging from 0 to 300 kg of N per hectare acre, and total N was applied over 3 splits. Here are examples to show how curves can be fitted to show the response of yield to N and calculate the economic N optimum. The economic N opt is defined as the amount of N necessary to achieve high yield with the lowest input cost while maximizing profits. This chart shows the yield response to N for the winter oat varieties at Carlow in 2019. The table is the N opt of 3 different trials, which shows that N opt can vary between locations and seasons. This is an example of the model being applied to a spring oat trial. As the three varieties had quite a different response to N, there was a statistical reason to calculate an N-opt for each variety. I don't want you drawing any major conclusions yet, but it gives an idea of how calculating N-opt can be beneficial, as no one wants to be spending more money on N if that does not increase the profit margin of the grain. These curves will be fitted to each individual trial to give us a clearer idea of any patterns over different seasons for varieties and locations. As I mentioned at the start though, in addition to yield, it is also important to consider grain quality, and high yields as a result of N doesn't necessarily mean high quality. Specific weight, screenings, kernel content, and hullability are traits included in Miller specifications. These results are from the 2019 trials at Carlow. The winter oats, which had 3 varieties, are on the left, and the spring oats, which also had 3 varieties, are on the right. The x-axis shows the different levels of N applied. These charts show that specific weight declined with increased N. The dotted red line represents the specific weight value that millers want, which is ideally 52 or above. So for a couple of the winter and spring oat varieties here, the milling quality specification is not met at higher levels of N. Screenings, the percentage of grain smaller than 2 millimeters, of all varieties increased with additional N. So in terms of these traits, too much N reduces grain quality. As seen here for kernel content, which is the percentage of groat to the whole grain, it doesn't change much between the N application levels, but does vary between varieties, which is more evident in the spring oat trial. Again, these charts show that for hullability, the ease in which the oats dehull, it is more stable across N treatments, but differs between varieties. The winter oat Griffin and spring oat Canyon are proving more difficult to dehull. These results therefore show that there were stronger varietal effects on kernel content and hullability than effect of N. To conclude, initial results show both treatment and varietal effects, often independent of one another, on yield and quality traits. It's important to emphasize that high yield does not automatically mean high quality. An optimal N to produce more yield may not be the optimal N to achieve quality specs. There is a large amount of data already gathered, 11 trials with another 4 this season, 5 winter and 3 spring oat varieties with over 20 traits measured for each. When all are integrated and analysed, the results will hopefully enable me to reach the aim of designing management practices to enhance both grain yield and milling quality of winter and spring oats, and an even greater goal would to be able to do this on a region-specific and variety-specific basis. Thank you for listening.
Thank you, Sara, for that. If you have any questions on aspects of Sara's talk presentation, please send them in through the Q&A tab on Zoom or through YouTube and Facebook, and we'll get to as many of them as we can in the Q&A session that's going to follow after Richie's talk. So for our next presentation, we're switching to beans, a crop of increasing importance. I'd like to welcome Sheila Alves, who leads our beans research, and the title of Sheila's talk today is Growing Field Beans in Ireland, Agronomic Practices and Current Issues. Thank you, Sheila.
Thank you, Ewan. Good morning, everyone, and welcome. We know that growing field beans as a break crop in cereal rotations has benefits, both in productivity and for the environment. For example, by breaking the cycles of pests in cereals and by reducing the N load of the overall rotation. But field beans also stand well on their own. With the current support, the crop itself represents a good margin. Moreover, field beans are high in protein and do not require the input of N fertilizers. However, the crop is relatively new in Ireland with significant production only from 2015. Therefore, we still have a lot to learn on how to grow the crop under the Irish agroclimate, the frequency and incidence of known and emerging pests, and their impact on yield and grain quality needs to be established. And also very important is to determine how to increase yield stability, especially in light of the climate changes. As many of you know, we did a survey last year aiming to capture the variability in yield observed in the 2020 season. The results in terms of yield are presented in this graph. We had a total of 81 values reported for yield, with an average of 4.7 tons per hectare, which is slightly lower than the 10-year yield average of 5.3. The variability in yield was huge, ranging from 1.2 to 7.6 tons per hectare. For comparison, in the seasons of 2019, we had an average yield of 5.5 and a range between 3.2 to 6.5 tons per hectare. So, what is the impact of that huge yield variability? What does it mean to be in this side of the graph with lower yields compared with this side of the graph, the higher yielding group? Let's look at those two groupings in more detail. This graph shows the output and margin in light green and red for three different yields presented in dark green. The first scenario is for the mean yield in 2020 of 4.7 tons per hectare. We have a margin of 251 euros per hectare. The second scenario represents the average yield for that lower 25% in yield. The value in average was 3.2 tons per hectare, and the margin is negative at minus 63 euros per hectare. The last scenario represents the average yield for the top 25%, with a value of 6.1 tons per hectare of yield and a margin of 545 euros per hectare. So we need to know what factors contribute to this variability, both at the environmental level, but also how can we improve the agronomic practices to increase yield stability. In terms of agronomic practices, we already have good research done that can guide in selecting the optimal seed, sowing and harvest dates, and disease control for Irish conditions. I'm going to highlight those in the next slides. So according with research performed in Oak Park, optimum sowing rates for spring beans should range between 35 to 45 seeds per square meter, with a negative impact on yield for values below or above that range. And in fact, most of the responses are in that range. However, a smaller percentage sown at lower rate, which with the dry spell felt in the summer of last season could be related with the lower yields. A note on varieties to say that 57% of the spring beans sown were lynx, effectively displacing both fuego and fanfare. Sowing date is one of the major factors that impact yield in field beans. Teagasc research shown that sowing after April brings a penalty in yields, and optimal sowing dates are between mid-February to the mid-end of March. This graph shows the reported yield values for last season, grouped by 4 sowing periods, with the highest yields observed between 15th of February and 31st of March, while sowing before or after gave lower yields. And in fact, in the last season, most of the spring beans were sown between the 15th of February and end March. Another known factor that decreases yield in this is disease. The most problematic disease through the last season was chocolate spot. Teagasc research has shown that the most effective fungicide program comprises of 2 applications, the first in early to mid flowering and the second at pot setting or pot filling. This is also supported by PGRO research in the UK. Earlier fungicide applications before flowering have been shown to be less effective in chocolate spot control. Furthermore, an early first application may lead also to a second early application, which leaves the crop exposed to late chocolate spot. So to summarize, opportunities for improvement are basically three. Avoid sowing below recommended seed rate, target a plant population of 25 to 35 seeds per square meter, avoid late sowing dates, specifically from April, follow recommended disease management practices. For chocolate spot, first application should be between early to mid flowering, second application between pot setting and pot filling. From our perspective, we'll focus ongoing and future research in identifying other sources of yield instability. We need to identify high versus low productive fields or regions. For example, sandy soils and drier areas may need a higher seed rate to compensate lower seed per plant. We need to minimize field losses for winter beans and increase resilience to dry spells, mostly in spring beans. We need to identify disease patterns for specific areas in view to design specific fungicide programs for that areas. This is all from me today. Thank you very much for your time.
That's great. Thank you, Sheila. Our next presentation switches focus to rye and is delivered by Richie Hacker. And the title of Richie's talk this morning is short and to the point, The Potential of Rye in Ireland. Over to you, Richie.
Okay, thank you, Ewan. So as Ewan said, I'm going to talk about our experiences with the rye crop, and mainly with winter rye we've worked with and growing it as a grain crop as opposed to a forage crop. So I suppose rye is still a minor crop in Ireland. So in 2020, there was less than 500 hectares of the crop grown, but indications are that that acreage has increased for the 2021 harvest. But the crop was grown extensively in some other crops in some other countries such as Denmark, Germany, Poland, and Russia. And it has a number of uses. I suppose the main use to date in Ireland has been for distilling, in the distilling industry for making rye whiskey. That's still a small market, but it's hopefully will increase over the coming years. But it can also be used in animal feed, particularly for pigs and ruminants. And my colleague Padder Lawler in Moore Park has looked at the crop as a potential feed inclusion for pig diets and has found it to be quite a good crop or grain to include in certain pig diets. It can also be used for human consumption. So many people will have heard of rye bread and rye vita, which is made from rye. And I suppose it can also be used as a whole crop forage for animals. But as I said, I'm going to talk mainly about the crop being as a grain crop. So why would we think about growing rye? What are the advantages of the crop compared to other crops? Well, I suppose the first one is that rye has a very high yield potential. So these are the results of some trials over the past 2 years, 2019 and 2020, where we compared a number of rye varieties in terms of yield. And in each experiment, we included 1 or 2 wheat varieties for comparison. And what I've presented here is the relative yield of the wheat in the solid bar and the rye in the hatched bar. And how the two crops compared. And you can see that the rye compared very favorably with the wheat, at least as good as the wheat in all the trials. And particularly for the 2020 trials, the conditions were less than favorable for the rye. But it has a number of other advantages. It has very good disease resistance, particularly to foliar diseases. So the main diseases that it's susceptible to are mildew and brown rust, and they're the 2 diseases you would have to take into account in designing a fungicide program for the crop. But other than that, it has very good disease resistance. It is also very good resistance to take-all, so second only to oats in terms of take-all, and that makes it a good option in situations where take-all would be an issue for a crop like wheat or barley. Rye has a very extensive rooting system, and that confers very good nutrient efficiency and very good drought tolerance to the crop. So it's able to scavenge the soil for nutrients and water better than other crops. And that's obviously an advantage in current times when we're experiencing periods of drought more regularly perhaps than in the past. It also has the potential to spread the workload, particularly around harvest time. So the crop will ripen generally between winter barley and winter wheat. So it gives you the option to spread your workload a little bit. And during the season also, rye would be more advanced in growth stage than say wheat. So that gives you the option to spread your workload during the season as well. Rye also has a high straw yield. So this is just a picture from one of our trials last year in Knockbeg, where we have wheat on the right, on the left, and rye on the right. And you can see there's a much bulkier sort of straw after the rye. I would say this is probably an exceptional year in that there was quite a bad drought at this particular site, which affected the wheat more so than the rye. But typically, from commercial practice, growers are reporting getting 1 to 2 4x4 bales of straw extra with rye compared to a comparable wheat crop. But the crop also has some challenges. It's not risk-free. So it's quite a tall crop, so it's 30 to 50 centimeters taller than an equivalent wheat crop. And that means that lodging is a risk. And lodging control will be an important part of any management regime. And I suppose that requires careful composition of 2 management inputs, nitrogen, and it will also need a robust growth regulator program. And that they're going to be key requirements of managing the crop to prevent lodging. But in terms of lodging, it probably should say leaning more so than lodging. So the crop tends not to go flat on the ground, it tends to lean over. And that's because it's got quite a strong straw. And most of the problems occur because of root roll, where the whole root system turns in the soil. And the crop does make every effort to, when this occurs, the crop does make every effort to upright itself again. We can see in the top right-hand picture, the node that's circled in white where the crop has tried to straighten itself up again. And generally at harvest, if the crop is leaning or lodged, it will slow down the combining operation, but you will get most all of the crop. You tend to have relatively low losses even in crops that are somewhat lodged. I suppose the other challenge is that market is currently limited, and there are also limited pesticide approvals for the crop. So not all products are allowed on other crops are allowed on pests are allowed on rye at the moment. That problem may well disappear if the amount of the crop grown in the country increases. In the past, ergo was a problem, but modern hybrids have significantly reduced the risk of that problem, as such that in the 2 years that we've worked with the crop in Oak Park, we haven't seen any ergo on the crop. It's also quite susceptible to slug damage, and I'll show a picture of that in a moment. And I suppose the other big limitation is that there's limited agronomy research under our conditions. We're relying on work from abroad to guide our management recommendations, and that's something that we need to rectify. I suppose in terms of the slug damage, this was from a very high-risk slug, high-risk situation last autumn, where we both rye and wheat sown on the same day at roughly the same seed rate. And you can see on the left, the rye was quite badly damaged compared to the wheat. But the plants are still there, so the slugs continue grazing the crop over the winter period, but the plants are still there, and the crop recovered afterwards. So I suppose in conclusion, rye shows good potential, but markets do need to be developed and increased for the crop, and the agronomy of the crop needs to be optimized. And I suppose just on that, we have a PhD student looking at the agronomy of the crop, funded by the Department of Agriculture. So we should have a lot more information on the crop in a few years' time.
So thank you, Richie. I'd like to welcome back Sheila and Sara now so that we can take some time to get through as many questions as possible. Before that, I'd just like to apologize. We had a couple of quirks with Zoom this morning, so I understand it's been resolved now. So hopefully everybody is able to connect in as well. So I'd like to introduce Michael Hennessy, who is the head of Crops KT here in Oak Park. Michael would share this session that for the questions that we've been getting in over the last few minutes. So over to you, Michael.
Thanks very much, Ewan. Look, we're going to get into the questions straight away. Sara, I'm just going to come to you first. We have a question in there about nitrogen timings in oats. Does it matter? And how many splits should farmers put on?
Okay, thank you for the question. So I'm not looking at timing myself, but in line with some of these response trials, which are part of a greater project, they do also have timing trials going on as well with AHDB. So it will be interesting to find out if timings do have a significant effect. But in terms of myself, I'm not looking into it.
Okay, thanks, Sara. Sheila, I just want to come to you for a second there. There's a question in here around winter beans. How much variation is in winter beans and how does that compare to spring?
That's a very good question. The winter beans in Ireland occupy a much smaller acreage than the spring beans, but our research shows that the variation in yield, at least due to the dry spells, is much lower in winter beans, and winter beans tend to yield higher too. So they are a good option. Okay.
Richie, I'm going to come across to you. Question on rye here. In terms of the very straw high yield that you mentioned, what sort of K offtake is that, or is that typical as you would expect it in other cereal crops?
Again, it's one of the questions we don't have an Irish answer for, but it's going to be a little bit hot because you have higher yield and K offtake is going to be linked to your straw yield. It will be higher than, say, wheat or barley. Exactly how much higher, I can't give you a definite answer on that.
Okay, and a really quick one, I suppose, really Richie, for you as well. In terms of harvest date, how does the harvest date for rye compare to winter wheat?
It'll be before winter wheat. It'll typically be between that of winter barley and winter wheat, between those two.
Okay. Sara, quick one back to you again. The relationship between Cs per meter squared in oats and the quality parameters. What's the relationship there?
Okay, so these are things I will be looking into. So at the moment, there's some positive and negative relationships between yield components and quality, but hopefully once it's all analysed with me, I've got another 1.5 years left, but hopefully I'll get another chance to show you those results.
Okay, great, thank you. Sheila, I'm going to come back to you for a couple of questions. In terms of the margins that you presented there to us, was land rental included in that? And maybe you might also have a comment in terms of the protein payment and how that would affect the margins as well.
Can I call Kieran Collins to help me with this? Because he actually gave me help to make these calculations. Kieran?
Yes, Sheila.
So did the margins...
Land rental is not included in those figures, so it's from the Teagasc costs and returns. So that would need to be factored in, as it would need to be factored in for all crops. Secondly, on the protein payment, yes, it's huge. It's hugely important in terms of the margins in beans, and I suppose without it, beans will struggle against other crops, particularly like the spring barley.
Okay, thanks very much, Kieran. I was just going to ask you just one very quick one then, Sheila, in terms of chocolate spot. Is there any other fungicides being trialed for chocolate spot in Ireland? Anything new?
Not that I am aware, no.
Okay, not so far. Okay, Richie, I'm going to come back to you in terms of rye here again. In terms of ergot, you mentioned earlier, how much of a problem is that? It used to be a problem in the past. Is it really that much of a problem now?
It can still occur, but most of the hybrid technology has works such that it produces the crops produce a lot of pollen, and the crop. So ergot gets into the crop during flowering, If the pollen grain doesn't get into the flower first, the ergo can get in, but a lot of the hybrids have a huge production of pollen that gets into the flower quite quickly, so it reduces the risk. There is still a risk, and you'd be advised to rotate your rye crops and to avoid sowing them in where there's a lot of grass weeds and things like that. But it is a risk, but it's quite a small risk in current hybrids from what we can see.
Okay, and a very quick one, Richie, in terms of protein, has rye much protein, or is it a protein source?
It would be somewhat similar to the other cereals, maybe a little bit lower than wheat. We haven't a lot of work again done on that, but it seems somewhat similar to wheat.
Okay.
So it's a very similar grade to wheat and barley in terms of its composition.
Okay, thank you. And just, Sara, I'm going to come back to you there for a second. In terms of the quality aspects for nitrogen, it seems to have quite a large effect. How much is it? I mean, can people really overdo it?
Yes, definitely, especially from some results we're seeing so far, especially with just too much nitrogen does reduce the quality, and this doesn't meet the milling specification. But it's kind of hard to define how much yet, because once we have a better idea of the relationship between all these traits, hopefully we'll get a clearer idea of how these traits then respond to N as well.
Okay, thank you very much. Richie, just coming back to you then for a second on the rye end of it. In terms of being susceptible to sprouting at harvest, how does rye stand up?
Quite well from what we've seen. So this year we had a site where we'd wheat and rye on it, and it was an outside site, so it was one of the last ones we got to harvest, and it got both of those big storms that occurred in August, and the wheat was badly shed and badly sprouted, but there was very little shedding and no visible sprouting on the rye. So I'd say it's quite it's not very susceptible to sprouting, but I would caution that adds a potential second problem, if you like, in that if you lose heads during the harvesting process, that grain tends not to germinate very easily, and it can lead to volunteers in the next crop. So one of the things I think we would suggest is not growing winter cereals after a rye crop. You either leave the ground fallow or sow something like oilseed rape, where you can control the volunteer rye plants.
Okay, and a very quick one then, Richie, just in terms of a question in here for somebody who wants to grow rye in the west of the country. Is there any potential problems there?
No, I suppose maybe lodging pressure might be a little bit higher in the west, maybe more rain, wetter soils, allowing more root roll on that. But I don't see any other reason why you couldn't grow it in the west.
Okay, maybe just one final question, I think, is all we have time for. And Sheila, I'm going to go to you. Just in terms of spring beans, are they more stable yielders when they're sown earlier in comparison to sown late?
The penalty in yields of sowing late and I'm talking about after April, relates with the time that the crop doesn't have to actually grow and have a big source to the seed, to give to the seed. So the crop will also harvest later, which has again a penalty. So sowing earlier will give you more stable and higher yields.
Okay, thank you very much, Sheila. So look, we have a lot of other questions. There's a huge amount of questions coming in. So what we're going to try and do is try and answer a number of these questions through typing them in the answers, so you'll be able to see some of the answers to the questions that are after coming in. So for the moment, thanks very much to Richie, Sheila and Sara for this section. I'm going to hand you back to you and now for the next section.
Okay, thanks, Michael. And it is great to see the questions and the feedback coming in. So, okay, we now turn to the second theme of this morning's session. We're all fully aware of the ambitions of Europe's farm to fork strategy that was published in 2020. And while the practical details have yet to be ironed out and the logistics of it, is clear that in the years out to 2030, there's going to have to be a greater focus on IPM-based approaches in our tillage systems to reduce the inputs that are currently being put on. So on that theme, our first speaker of the second part of this morning is Max Shugart. Max is a Walsh scholar here in Oak Park, completing his PhD under the supervision of Louise McNamara and Michael Gaffney. And Max is completing a very important work on establishing a BYD and aphid monitoring network to support cereal-based IPM strategies for the tillage sector. Thank you, Max.
The grain and the birch cherry oat aphids are both main vectors of the barley yellow dwarf viruses. Both the EU ban of neonicotinoids and pyrethroid-resistant grain aphids, which we call the SA3 clone, impede control with insecticides as well. In addition to that, we have evidence for metabolic insecticide resistance, which might challenge BYDV control in the future. My research is about the virus transmission capability of the resistant SA3 clone, which is currently unknown, but also on the impact of migrating aphids towards the BYDV spread, where we don't know lots of at all, let's say. So to target and catch migrating aphids, we have a suction tower network set up in all over Ireland. We have a tower, a 12-meter tower in Oak Park, which is running since January. And we have just set up a second tower in Cork, and a third tower in Ashtown is going to be set up. And those towers, they target migrating aphids from a certain radius, and those aphids we can test for BYDV and insecticide resistance. So we have several advantages of the suction tower network. First, we can look for aphid migrations and link those with weather data. Second, we can have a look at the knockdown resistance and see to target how many of those aphids are insecticide resistant. And third, we can look at the BYDV transmission to investigate the impact of different migrating aphids species towards BYDV spread. So at first I want to start with some data from the aphid migration of the suction tower from Oak Park in 2020. On the left side, we have the number of aphids caught towards all over the months and weeks. You can see that the aphids, they start to show up between April and May, and up to June, we get higher numbers. Between July and August, we caught many Essevina. We stopped catching them at the end of August, but we got high numbers of Alpada then with a tiny drop, and then the second high peak towards September, October, and November. If you want to have in mind that your winter cereals are at risk between, if you saw them between September and October, I want to point out that two things. First, that by biological reasons, the Arpadae, they migrate to their winter host, the cherry tree, between September and October. And the second thing is, we don't know whether those aphids land in the crops and spread virus, and we don't know the virus load of those aphids. So that's also part of my investigation for my research project. A second thing which we can do is to link aphid flight with weather conditions, as I said earlier. We know that the aphids start flying at temperatures around 15 degrees, and we can see that if we get mean temperatures above 15 degrees, aphids flight numbers get really high. In August, for example, where we went over like up to 17.5 degrees, we got high numbers, and then it dropped down and it got up again with really high numbers of Alpada, and then towards the end of the year, numbers fell. I want to raise your attention towards July, August, and September, where we had Storm Ellen, if you might remember, this year, where we had temperature drop and also low aphid numbers. We are able to not only link migrating aphid numbers towards temperature, but also towards wind. Over here, we had Storm Ellen, which really high maximum wind numbers and low aphid numbers. On the other hand, we had low numbers, low wind, and high aphid numbers towards the beginning of August, and in the beginning of September, we had the same thing. We can also do the same with rain data. And as you see, with low rain, we get high aphid numbers. With loads of rain, we get low aphid numbers, and then vice versa again. with low rain, we get high aphid numbers again. However, this is the first year data, but it also shows us some interesting things. We basically know that the numbers of aphids vary between the weeks of different weather conditions, and that the most of the aphids start flying in spring at mean temperatures above 15 degrees. We know that we saw that migrating Asavine are found until mid of August and migrating Arpada until the end of November. However, most of those most of the numbers declined until the end of October. And we also got that strong winds and rain negatively impact aphid flight. However, the data from all of the 3 suction towers, which we are going to be set up, will of course improve our resolution over the years. So we will be able to do accurate predictions, hopefully in the future. The second thing I want to talk about is I've been testing those aphids for the knockdown resistance, which makes the aphids, the Asavine aphids, partially resistant. In 2017 and 2018, we found 25 and 20% of the Asavine tested positive for KDR. The migrating aphids from the Oak Park suction tower, we had numbers of 22%. We also had a second suction tower up in Ashtown, only a 6-meter tower, but still, where we found 18% of those aphids partially resistant, leading to the conclusion that still the partially resistant aphids are widespread. I've been also testing those aphids for homozygous resistant superclone, which I couldn't find, which is good news for us. Last but not least, I want to talk about a new technology which we just established at Teagasc. It's called the Digital Droplet Pcr. It basically allows us the quantification of BYDV copy numbers. It can help us in the detection of low virus copy numbers, either within positive aphids or in symptom and asymptomatic barley leaves. For example, if we have a barley leaf and it's yellow, we mostly know, okay, barley yellow dwarf virus and it's too late. However, you can also have loads of viruses in asymptomatic leaves. We currently have this lack of knowledge of virus levels and which are in the field, and hopefully the digital PCR allows us to determine those virus level and so we could use the digital PCR as a kind of early diagnostics tool to hopefully assist future IPM approaches in the future, which would be totally awesome, and I'm super excited to do this. So to sum it up, outlook for 2021, I want to do loads of fieldwork, and the suction tower sampling will of course enhance our knowledge on BYDV spread and insecticide resistant aphids. Hopefully the digital PCR will improve the early detection of BYDV levels, and all of this together, the prediction of the occurrence of BYDV and insecticide resistance aphids will assist our future IPM approaches. So I want to thank all of the guys from Teagasc, which have been helping me a lot during this my first year. Also my supervisors from UCD, Maynooth University and INRE, and also shout outs to all of the Walsh scholars. And yeah, with that, thanks for your attention and goodbye.
Okay, thank you, Max. Very interesting. We look forward to hearing how the network of towers and the diagnostic capacity develops in 2021. Our final speaker this morning is Dan Milbourne. Dan is the PI of the DAFM-funded Vicky project, which has recently concluded and made significant advancements in regards to developing genetic resources to support variety development. So in this vein, the title of Dan's presentation this morning is Producing High-Value Plant Breeding Materials Suited to Irish Tillage Systems. Thank you, Dan.
Thanks, Ewan. So we've been talking in this session about disease control. And if you look at approaches like integrated pest management, shown here for blight control, we can see the different components required to make it work. And the one I'd like to focus on today is improved varieties. And more specifically, I'm going to talk about how research in Teagasc is contributing to breeding the varieties that we need for these types of sustainable production approaches. So plant breeding is a really powerful tool, but it suffers from a major drawback in terms of the timescales taken to deliver the final product. So from the initial cross-pollination of parental material to produce breeding populations, it can take it from anywhere between 7 and 10 years to select a better performing variety. And this is due to the necessity for things like seed propagation and multi-year trialing. An additional problem is that combining multiple desirable characteristics is frequently only possible over several breeding cycles. And this makes the achievement of some breeding goals a multi-decadal process. So today I'm going to give you 3 examples of work at Teagasc where we're trying to address these problems and speed the release of varieties for Irish tillage systems. The first example I'd like to show you comes from the Teagasc IPM potato breeding program. And here I'm actually going to show you the endpoint of a breeding cycle and tell you how we got there a lot faster than we might have. Here's the recently released variety Buster. And as well as its excellent yield, one of the things that we're really excited about is that it's extremely resistant to both major species of potato cyst nematode or eelworm. So here are the results of a field trial commissioned by IPM in the UK, where Buster and Maris piper were planted in nematode-infested soil. The blue bars show how many eggs per gram of soil were present at the start. The trial wasn't treated with nematicide, and you can see that the infestation level of Maris piper by harvest time, and that's the orange bars, is very high, whereas PCN levels have actually dropped where Buster is planted. So Buster protects its yield potential better, and it even might actually reduce the nematode levels in the surrounding soil for subsequent crops. Buster is resistant because it's got 5 PCN resistant genes that were combined from multiple parents during the breeding process. And we achieved this using a genomics approach called marker-assisted selection, which allowed us to more precisely identify which parents to combine. And this sped the developmental process up significantly. Normally, this combination of genes might take up to 3 cycles of breeding to accumulate, and that's about 30 years. But marker-assisted selection allows us to do it in a single 10-year cycle. And we now routinely use marker-assisted selection in the potato breeding program to develop disease-resistant varieties for various pests and diseases, including PCN, blight, viruses, and wart disease. The second example I'd like to show you comes from the recently ended project Vicky, which was funded by the DAF stimulus program. In Ireland, the ideal bean variety is one that can give high yields from autumn sowing, but it's got good resistance to protect this yield in high disease pressure years. You could argue that varieties bred for Northern Europe and the UK don't really fit the bill here. And we wanted to address this by creating a pool of material that would be suitable for breeding Irish adapted varieties. So now, whilst the breeding cycle in the commercial program can take up to a decade, under certain circumstances, it's possible to perform 1 or even 2 rounds of crossing and selection per year to rapidly improve individual target traits. This is an imperfect process, and it doesn't produce varietal material, but it can establish a pool of breeding material from which future selections can be performed. So our collaborators in the UK assembled a starting pool of material, including elite spring varieties and lines exhibiting resistance to chocolate spot and ascochyta. And we performed 5 rounds of crossing and selection over 4 years, including 2 rounds in the field conditions in Ireland, trying to combine yield under disease pressure. We call this rapid cycle breeding. At the end of the process, this material was trialed with elite varieties at Oak Park, and this shows the plot yields of the 2 field selection years in Ireland relative to 2 of the parental varieties, Fanfare and Vertigo. The selection performed in 2018 actually looks quite promising, and we're assessing the possibility of using it to develop autumn-sown disease-resistant varieties, although this will really require further assessment and might also involve further rounds of crossing and selection. The final example I'd like to show you is also partly related to the Vicky project. Septoria resistance in winter wheat is an important trait in Ireland, but perhaps less so in countries where many breeding programs have their major markets, so it's not necessarily a priority trait for these breeders. In Vicky, we work with partners on identifying Septoria resistance in populations that hadn't previously been characterized for resistance. And here's an example of resistance in the wheat magic population developed at NIAB in the UK by intercrossing 8 elite varieties. There are 1,000 lines in the population. You can see in the results from this unsprayed trial that there are individuals showing lower levels of septoria or higher resistance than the parents. This allowed us to understand the genetics of resistance. And it's not inconceivable that some of these lines could be used for varietal development, although there'd still be quite a long lead time for that. One interesting consequence of Vicky is that during the project, we developed stronger relationships with commercial wheat breeders who were interested in our experimental results and excited by our capacity for large-scale testing for septoria resistance. So subsequently, we started collaborations with these companies to test their advanced breeding material at Oak Park. And here's an image of one of the recent trials. So importantly, the material being tested here is from the later stages of programs, but before the varietal selection has been performed. So through this collaboration, breeders are gaining better information about the characteristics important in Irish production. And that can feed into their varietal selection decisions. These sorts of collaborations increase the possibility of these new varieties having better targeting, and we're seeking to expand on them in the future. So I hope I've shown you that although breeding can seem frustratingly slow, at Teagasc we're really focused on developing solutions to reduce the timescales and increase the targeting of varieties for Irish tillage systems. The solutions we're adopting vary from case to case depending on the situation and resources already available for that crop. So, for instance, where we have control of the breeding programs in potatoes, we can develop genomics approaches to increase speed and precision. In situations where we don't have a breeding program, such as in beans, we can create the conditions for subsequent improvement efforts by a concentrated burst of pre-breeding activity to produce breeding material that brings us closer to the ideal variety. And finally, as I talked about for wheat, developing these mutually beneficial collaborations with commercial breeders to work on important traits for Ireland in pre-varietal material is a really promising avenue which we hope to expand on. And finally, I just want to mention that this is really just a snapshot of the types of work we're doing to improve plant breeding in oat parka. We have other work on things like using genomic selection and speed breeding, and we're also maintaining a capacity for approaches such as gene editing, should that be needed in the future. So finally, once again, I'd like to acknowledge the Department of Agriculture, Food and the Marine for funding the Vicky project, acknowledge the partners of the Vicky project, and on the right, I'd like to acknowledge the people who carried out the work from the examples I described today. Thank you, and I'll hand you back to you.
Okay, thank you, Dan. It really is clear that enhanced genetic resilience is going to be key to meet these farm to fork goals. We'll now take some time for questions on the last two presentations, and as they're coming in, we're still actually answering via the chat some of the questions from the first session as well. So I'll pass back to Michael to field the questions for Max and Dan, please.
Okay, thanks very much, Ewan. There's a lot of questions coming back in here again, so we'll try and get through them as quick as we can. Max, I'm going to come to you first. Max, just in terms of the wind effect and its ability to either stop or the inability maybe of our tower to pull in those aphids, is it a fact that if the wind is there that the aphids aren't flying, or our tower can't actually capture them?
To be honest, this is the first year data, so we don't know exactly about the wind effect. So what we know is when we have strong winds and loads of rain that the aphids don't fly, like biological. I don't know if the tower won't suck up any aphids. It's most likely that they just don't fly at strong winds. So that's it.
Okay. And I'm going to ask you another one just in terms of BYDV again, and maybe Ewan might want to chip in here as well. In terms of BYDV in an infected plant, is the virus uniformly distributed to the plant, or is it mostly concentrated in the yellow parts of the leaves? Do we have an answer to that yet?
Yeah. The virus can be basically everywhere in the plant. So if you only take a tiny part of the plant and do an extraction, it's likely that you catch up loads of aphids or not. So what we usually do is take the whole plant and mash it up, and then take some of the of all of this mashed up material, take some extracts and do our experiments with that.
And for the new tool that we have, Max, is it the case where that can detect whether there's virus in that plant even when we can't see yellowing? Is that the case?
Yes, exactly. So it's a tool to like an early detection kind of tool. So we can detect if we have super tiny virus loads in our plants, we can detect those without seeing symptoms in the plants. However, we just got this technology in, so this is theoretical, and we hope that in the next few years we can assist in IPM approaches with that. But right now it's too early to say that.
Okay. Dan, just a quick one there for you in terms of the stacking, if you like, of the various different traits that you're putting into varieties. Is some of the newer varieties, Buster for instance, is that totally resistant to or tolerant, I'm not sure which, to both types of potato nematode?
Yeah, so there are two main species of Globidera of the golden potatoes nematode. There's Globidera rostokiensis, and Globulera pallida. And Buster is resistant to both of those. One of the genes actually provides complete resistance to the major pathotypes of Globulera rostokiensis that we find in Northern Europe, including Ireland. And the other 4 genes, they're combining to provide resistance to the most prevalent forms of Globulera pallida, which theoretically we don't have much of in Ireland, but it's providing sort of protection from that perhaps coming in and expanding, which has been the experience in the UK, that once rostokiensis populations go down, pallida populations go up.
Okay, and then I might just follow up, just ask you another one in terms of blight, which maybe more people might be more familiar with. Just in terms of stacking in genes for resistance to blight in potato varieties, how are we there yet in terms of some of the Irish varieties that we have? And if we are they going more suitable towards organics or much less fungicide use for blight control?
So surprisingly, we're a little bit behind in blight resistance than we are in nematode resistance. And most of the varieties that are available at the moment, and there are some very good blight resistant varieties, but they're based on single R genes. So for instance, the Teagasc variety Java, which was recently released, is very, very resistant to blight. But that was developed through a similar process of marker-assisted selection. But we don't have a lot of availability for until recently for lots of different wild genes that we could stack. So that's sort of what's going to be the next generation of resistant varieties, where we take multiple R genes and use the approach that we used in Buster to rapidly stack those into varietal material. And the real point here is that we need to use IPM approaches to protect the resistances that we've got now, to increase their lifespan, and then we'll have the time to develop this second generation of varieties, which will then contribute even further to the sustainable types approaches that we need to start using.
Okay, thanks, Dan. I'm going to go back to you, Max, and maybe you might maybe chip in here as well. In terms of BYDV, and we now know a bit more about perethroid resistance. Is there going to be any revised differences, if you like, in agronomic recommendations for 2021?
No. Right now, like I just started my second year. We only have one suction tow running. We don't have we're just currently setting up the digital PCR. So right now it's definitely way too early. So I still have 3 more years in my PhD. Hopefully we can tell more about that afterwards. But right now it's definitely too early.
And I can just add to that, Michael, with the second tower turned on as well, it gives us greater coverage. And obviously we have a third one and possibly more in the pipeline. But definitely the more information we have, the greater confidence we can put into if we need to make any changes to any advisory recommendations. But that's the main reason the network is established in digital PCR as well, is that there are unknowns there that we need to address. So, and that's important for the sector.
Quick one, Ewan, just back to you again, just for a second. There's a question there about the location of the towers and their proximity maybe to big towns or large cities. And is that still going to be relatively a good position for them to capture data from an area outside, you know, and maybe within a distance from them?
Yeah, no, it's a valid point, but the other thing is that, and we haven't touched on it today, is we have a series of mobile towers as well, that we can install pretty much anywhere, and that network will be expanded as well. So there's actually going to be 3 tiers in the sampling: the 12-meter tall towers, we have the 6-meter towers, and then there's also more focused sampling that can be done in fields as well. So it'll be a multi-layered, but yeah, it's a valid point. There's a balance between logistics because these things have to be collected every week. And also obviously aphids are important for the horticultural sector. So that's the other reason why Ashtown was selected, but definitely up towards the northeast, there is something there as well that we will be looking at.
Okay.
Maybe I could add to that, that as well, these suction towers, they're 12 meter high, and they basically, we're not sampling aphids from the field, but aphids who are migrating and going with the wind. Like they take off and get taken away with the wind. So we have a range from literature. It's around an 80-kilometer diameter around the tower, from which we can basically sample aphids. So it's not really necessary to have a field right underneath it.
Okay, thanks, Max. We're just gone a little bit over time, but we have a good few more questions, so we're just going to keep going for another 5 or 10 minutes. I hope everybody can stick with us. Dan, I just want to come back to you for a second. A question about the PCN again, and is it the fact that the resistance that's in there, does it prevent the cyst from germinating and getting going, or is it from the feeding of the nematode afterwards?
That's a really good question, because one of the biggest control problems with PCN is that they insist and they can stay in the soil for years. We found cysts, viable cysts, you know, in over 10 years time. So and that makes rotations very, very challenging. So the resistance genes don't prevent the nematodes from hatching. Potato and other crops, you know, this is why we have trap crops that release things from their exudates from their roots, which induce the hatch. And most of the resistance genes, they act in slightly different ways for different genes, but the vast majority of them prevent the feeding of the nematodes. And really what happens is the females are feeding, and then they're not, the sexual cycle that produces the eggs for the next generation also isn't able to occur. This is it's actually really important. It's much more advantageous in an integrated pest management context to allow the nematodes to hatch and then for them to be defeated by the plant, because what that does is over time it causes the total nematode load in the soil to drop. And you could see in Buster, it wasn't statistically significant necessarily, but numerically there were fewer nematode eggs in the soil after Buster had been planted than before. And you can imagine that is a way of, in one sense, sterilizing the soil over multiple growing cycles.
So not only, Dan, is it good in the season that it's in, but for a long-term integrated pest management type of control.
Yeah, and that's a real key point. It's not just, you don't just get those in-season benefits using these types of varieties, but it's important to put that in the context of a wider IPM management strategy, because you've got to protect any one pillar of IPM with using the other pillars of IPM.
Okay, Sheila, I see you there. I'm just going to come across, and there's been lots of questions on beans, and I'm just going to ask you a quick question there on that. It's in terms of sowing depth and its subsequent effect, perhaps on pod height or pod height on the plant as it grows later on. Any correlation there?
I was actually answering to that. No, I'm not aware of any correlation between sowing depth and the pod setting in the plants. I mean, the pod setting the plant depends on the canopy development, which depends on seed rate more than anything else. And then, of course, the weather conditions and so on.
Okay, thanks, Sheila. I'm going to throw this one out as a question to both Max and maybe Ewan. There's a Syngenta app out there at the moment, which I suppose relies on the temperature in fields and correlates that with the sowing date of the crop, and does a bit of a calculation to give growers an indication of when the risk period is and when you may well get out and put an aphicide on. Any research as regards how that correlates to current advice? What way does that stack up?
No, go ahead.
Go ahead. No, go ahead, Max. Okay.
So basically, I cannot give you loads of input, so I want to transfer it to Ewan. But like, I'm looking for the migrating aphids mostly, so I cannot give you advice on that. So maybe Ewan.
Yeah, it's not something that is part of Max's program, but I think it's clear it's something that we're going to have to look at. The idea of the model that Max is developing is to again put those multiple layers of the environmental parameters onto it. But I suppose the other question as well is unknown is that while we have, Max touched on it in his presentation, is while we have asymptomatic plants in the field, they look green. The question is, do they have virus in them? And I suppose that's going to be the strength of the diagnostic platform that we invested in towards the end of last year. It's going to be allow us to answer those questions. So what's going to be very interesting is when we can try and correlate it with the likes of the apps that the companies have produced as well. So.
Okay, thanks very much, Ewan. Time is kind of against us, and I might just leave it with a final comment to everybody here, and to Max, to Dan, and to Sheila for answering all the questions that came over it. Thanks very much. You're very quick and honest answers. It's very difficult sometimes to come up with the answer quick enough. So look, thanks very much for that. We'll leave it there in terms of questions, and I'll hand back to you, Ewan.
Okay, so as we wrap up, I want to bring your attention just to the next in our series of webinars, which is the crop agronomy update on Tuesday the 9th, 11:30. This will provide updates on agronomic decisions to be considered over the next 3 to 4 weeks, and will cover crops across the country. And then part 2 of our tillage conference will be on February the 17th. Same time, 11:30 a.m. And we look forward to having you join back in for that, when we will hear research updates on septoria, grass weed control, and the results of a 6-year study on crop establishment systems as well. And of course, all of our crops, KT webinars, and research updates are available on the Teagasc Crops YouTube channel. So I want to conclude today's event by thanking all the speakers, Michael for chairing the Q&A, Dermot and Kieran answering questions via Zoom chat, Dara for managing production, and obviously yourselves for taking the time this morning to participate and tune in. So until the next time, take care and stay safe.